Hello everyone, and thank you for joining this presentation. Today we'll be talking about handling biomedical images and sharing them as reproducible workflows in accordance with FAIR principles. Our agenda consists of 5 points. First, understanding bioimaging. Second, planning and acquiring images. Third, gathering image metadata. Then, organizing and sharing image data. And finally, sharing reproducible workflows. Let's dive in. To begin, let's look at the broad landscape of bioimaging. Biological imaging plays a central role across multiple disciplines, from molecular biology and genetics to microbiology, physiology, and anatomy. Different imaging modalities serve different purposes. Electron microscopy gives us ultrastructural detail. Light microscopy allows us to observe living systems, and clinical or human bioimaging enables diagnostics and large-scale studies. But regardless of the method, the outcome is similar. These data that must be analyzed, managed, and interpreted. As this cartoon humorously shows, dealing with data can feel overwhelming, especially in the field of bioimaging. We've now entered the era of big data in imaging, with increasingly large and complex datasets being generated routinely. This brings great opportunities, but also major challenges. How do we handle and manage all this data properly? And how do we ensure our image analysis workflows are reproducible and shareable? The pressure of these questions is real, but the solutions start with adopting better practices in data stewardship. This diagram illustrates the life cycle of bioimaging data and the infrastructure needed to manage it. From image acquisition to processing, publication, archiving, and eventual reuse, each stage depends on robust tools, standards, and support systems. When handled properly using FAIR principles, bioimages gain new potential for scientific discovery, allowing reuse and reinterpretation. Let's see some fundamentals of planning imaging experiments and image acquisition. Before even clicking the shutter, we must ask, where will these images live? How will we move them, document them, and analyze them, not just today, but in the years to come? This image is all too familiar. Overflowing local drives, storage bars in the red. Conventional file-based systems can no longer keep up with the size and complexity of imaging data. That's why planning ahead is essential. Drafting proper data management plans, using scalable platforms like Omero, and adopting standardized new generation file formats, NGFFs, will become not just helpful, but essential. We must shift our mindset to long-term stewardship, not just short-term storage. Once imaging starts, data quality depends heavily on acquisition parameters. This includes proper instrument setup, calibration, and choices like image resolution, bit depth, and exposure. Think about capturing just enough detail to answer your research question without overwhelming your storage and analysis capacity. Always prioritize open formats like TIFF over proprietary ones like CZI. Open formats ensure better accessibility, long-term reuse, and compatibility with analysis tools. Importantly, don't forget metadata. Metadata is essential for interpreting the image. What instrument was used? What were the settings? Without this context, an image loses much of its scientific value. Data acquisition isn't just a technical step, it's foundational to reproducibility and FAIR data. What parameters should I use to answer my research question? This slide shows the exact same brain section captured with 2 different image bit depths, 8-bit on the left and 16-bit on the right. Notice how the 8-bit image is smaller, only 23 megabytes, but comes at the cost of dynamic range. The 16-bit image, while heavier, retains more intensity levels. Do you need a 16-bit image to answer your scientific question? It's essential to consider what imaging parameters are required or sufficient for each case. In other words, always balance what you need for your analysis with what your system can store and process. Microscopes are powerful tools, but their output is only as good as the person operating them. This is a reminder that no matter how advanced your imaging system is, the quality of your results ultimately depends on proper training, attention to detail, and a strong understanding of imaging principles. Investing time and training can significantly boost the reproducibility and reliability of your data. Microscopy images are often generated in proprietary formats like CZI from Zeiss or LIF from Leica. These formats can limit interoperability and long-term access. To ensure your data are fair and reusable, consider converting them to open formats like TIFF. One of the most widely used tools for this task is bioformats, developed by the Open Microscopy environment. It is integrated into FIGI/ImageJ, allowing seamless conversion and metadata preservation. Additionally, for more programmatic workflows, a Python version of bioformats is available via PyPI. This gives researchers full control over batch conversion, integration into pipelines, and compatibility with other data processing tools. OME-TIFF is a powerful open file format developed by the Open Microscopy environment. It's built on top of the standard TIFF format, but with critical enhancements for bioimaging. It integrates OME-XML, a metadata standard that allows rich annotation of image acquisition parameters, which are essential for reproducibility. OME-TIFF supports multidimensional and multi-image datasets, such as Z-stacks, time-lapse sequences, and multi-channel images. For large datasets, the Big TIFF extension allows storage beyond the 4 GB TIFF file limit, which is common in high-resolution microscopy. Finally, OME-TIFF supports file segmentation, distributing data across multiple files when needed. This is ideal for scalable storage and high-content screening. OME-ZAIR is the next-generation open file format for scalable bioimaging. It uses multi-scale image pyramids, like the one shown here, to efficiently store and display very large images. This allows fast access to different resolutions and slicing angles without loading the full resolution data each time. OME-ZAIR is compatible with many modern imaging tools, including FIGI, NAPARI, Neuroglancer, and Visar, as well as the 3D Cell Viewer from the Allen Institute. If you're working with batch image datasets, the community provides conda-based conversion tools, and you can handle OME-ZAIR in Python using the OME-ZAIR pyramid library. As a take-home message, strategic planning and data management, combined with the use of cloud-ready platforms and open file formats, are essential to ensure that imaging data is scalable, accessible, and ultimately reusable. Let's talk about image metadata. High-quality metadata is essential to make bioimaging data findable, accessible, interoperable, and reusable. Unfortunately, the current ecosystem is fragmented. We lack clear and consistent metadata standards, and both journals and institutions rarely provide sufficient guidance. These inconsistencies limit the sharing and understanding of research images. To address the fragmented state of image metadata, the scientific community has developed REMBI, Recommended Metadata for Biological Images. REMBI proposes a set of metadata guidelines that aim to satisfy the needs of research biologists, imaging scientists, and computer vision experts. This Venn diagram shows how their metadata requirements overlap to support interdisciplinary data, reuse, and reproducibility. A summary REMBI template is available in our GitHub repository for you to explore and use in your own projects. Here you can see the REMBI metadata template. It covers essential metadata modules such as the study context, sample information, imaging methods, acquisition settings, and analyze data. To ensure clarity and interoperability, it's important that your metadata contains controlled vocabularies, also known as ontologies. These standardized terms allow machines and researchers to unambiguously understand what organisms, genes, proteins, and methods were used. For example, use the NCBI taxonomy for organisms, UniProt for proteins, and FBBI for imaging techniques. Using tools like the Ontology Lookup Service from EMBLEBI or BioPortal, you can easily find controlled vocabulary terms for biomedicine, from cell types and experimental conditions to imaging modalities. You can explore definitions, check synonyms, and even find links to related terms, all in one interface. Tools like the Micrometa app can help you ensure accurate and complete metadata from your imaging setup. This application helps you document both the hardware configuration of your microscope and the settings used during acquisition, like magnification, exposure time, detector gain, and laser power. It provides a visual interface where you can drag and drop components, ensuring standard and consistent descriptions. The Methods J2 plugin for Fiji simplifies the writing of your microscopy materials and methods section. It uses metadata from a file created with a Micrometa app to automatically generate structured, standardized text. The output includes detailed information on the microscope, hardware, and imaging parameters. You can access the plugin from GitHub or explore its validation in the referenced article. Omero is one of the most powerful platforms for managing and sharing microscopy data. It extracts metadata from vendor-specific formats and organizes this information using standardized ontologies. With tools like MDIMIC, the metadata editor for microscopy, researchers can easily explore, edit, and export image metadata in structured formats, such as .txt, .tsv, and .csv. Omero is a fair compliance solution for storing, annotating, and reusing images. To make microscopy data reusable, we must go beyond just storing images. We need high-quality metadata and clear documentation. These two checklists from Schmid et al. 2023 outline best practices for image publishing. The first one focuses on image format and visual presentation, cropping, separating panels, adjusting brightness and contrast, and ensuring accessibility for colorblind users. The second checklist emphasizes annotation and availability, providing scale bars, explaining symbols, and sharing raw data using lossless compression through public repositories. To truly make bioimaging data fair, we must move toward three essential goals. First, we need better consolidation of metadata standards. Today's landscape is fragmented, and harmonized guidelines are critical for consistency and reuse. Second, we need to develop better tools for automated metadata collection. This requires working closely with microscope manufacturers to ensure metadata is accessible and understandable. And third, institutional and national research infrastructures must provide strong support for metadata recording, from planning to publication. Our next topic is the essentials of organizing and sharing image data. Imaging data often lives in many places, from personal laptops used by students and postdocs, to local institutional networks, to cloud storage solutions like Google Drive. But biomedical image data can easily reach the scale of terabytes or even petabytes, far beyond what these solutions were designed to handle. This makes infrastructure, workflow optimization, and standardized sharing protocols essential. Without these, image data becomes siloed, fragmented, and difficult to access, or worse, lost. When selecting a storage solution, we shouldn't ask, "Where can I save this?" We should ask, "How can I make this accessible and usable for others and for myself in the long run?" This is where the FAIR principles come in. We want our imaging data to be findable through persistent identifiers and indexing in searchable resources, accessible with open formats and clear software requirements, interoperable thanks to rich, standardized metadata, and reusable through descriptive documentation and clearly stated licenses. So, where should you share your bioimaging data? Definitely, there's no one-size-fits-all solution. It depends on your data type, discipline, and institutional policies. Some generalist repositories that support fair image sharing include OSF, ideal for collaborative projects; Dryad, used in life sciences; FRDR/DFDR, tailored to Canadian researchers and big data sets; BioStudies, designed for biological imaging and omics; Zenodo, integrates with GitHub and supports datasets, code, and publications; and Borealis, a Canadian Dataverse instance with institutional support. This table, from Schmid et al. 2023, outlines key features from different repositories such as file size limits, costs, whether they accept non-image data, and the type of metadata they require. Platforms like Zenodo and Dryad support a wide range of research outputs, while others like the Bioimage Archive are tailored specifically for imaging datasets. While the metadata requirements differ, it is a good practice to provide well-documented datasets to allow their understanding and reuse by other users. Now let's turn to two specialized repositories for bioimaging data. On the left, we have the Image Data Resource, IDR. It hosts curated datasets from published imaging studies and applies a standardized metadata model to ensure reusability. On the right, the Bioimage Archive at EMBLBI also supports datasets from diverse modalities, aligning with FAIR and REMBI principles, and working in tandem with IDR and MPR. Beyond public repositories, there are infrastructure solutions for local or institutional use. Omero is a secure server platform for managing and sharing microscopy data. It supports over 150 file formats and offers access via desktop or web. iRods is a rule-based data management system that provides advanced features like automated ingest, metadata indexing, and storage tiering, ideal for core facilities or research institutions. When sharing imaging data, quality and transparency are key. Consider the following. Share both raw and processed images, but make sure processing doesn't overwrite or alter the original data. Use uncompressed or lossless open formats like OMI-TIFF to retain original metadata. For annotated visuals, PNG is preferred. Always adopt clear, machine-readable naming conventions that reflect grouping factors like subject group, area marker. Include a README file to describe the dataset and the methods used. Finally, use open licenses such as CC BY or CC 0. As a reminder, data stewards strongly discourage making images available only upon request. This puts the burden of sharing on you indefinitely, which reduces your continued capacity for fair research. Sharing by depositing in a repository means you do it once, and everyone, including yourself, get to enjoy persistent access to the dataset with less effort on your part. Finally, let's talk about sharing reproducible workflows for research outputs. In image-based research, the analysis must meet three essential criteria. First, must be as objective as possible. Methods and measurements should be free of bias and based on clearly defined parameters. Manual counting or measurement should be avoided at all costs. Second, it must be reliable, producing consistent results under similar conditions. And third, and perhaps most importantly, the analysis must be reproducible so that others, or even your future self, can repeat the analysis and reach the same conclusions. Achieving this requires transparent documentation of workflows, version control, and accessible tools. From a research data management perspective, image analysis in biomedicine must follow a structured and transparent process. First, large, complete datasets must be accessible, ideally in open formats. Second, we need clear records of image processing steps, whether it's raw code or scripts or executable pipeline files. This ensures traceability and reproducibility of research outcomes. Third, we should be sharing not just the raw data, but also elements such as processed images, graphs, or figures, and related data tables. Finally, everything should be accompanied by clear naming conventions, README files, and metadata. With a modular pipeline, we can break down image analysis into distinct, manageable tasks. In this example, the pipeline starts with nuclei detection, followed by quantification, and ends with spatial analysis. Each step operates as an independent module. This approach allows researchers to build complex workflows by combining smaller, well-defined components. The advantage is that these modules can be designed and executed independently across projects, making the process more flexible and scalable. In image analysis workflows, we can identify a hierarchical structure, moving from technical image processing to biological interpretation. On the low-level side, we perform tasks that require technical expertise, such as pre-processing, segmentation, or object tracking. These steps generate data, but don't yet provide biological meaning. Then, on the high level, we apply disciplinary knowledge to interpret the outputs through statistical analysis, visualization, or modeling. This is how we transform raw microscopy data into meaningful scientific outputs. This diagram shows 3 common types of image analysis workflows in biological research. On the left, we start with microscopy images. In the middle, we see 3 possible analysis paths. First is an established workflow that uses a predefined tool or plugin. The second option is a custom workflow built by chaining together scripts, macros, or plugins. While this approach offers flexibility, it requires a certain level of technical skill. These skills must be considered foundational for researchers, not peripheral. A third approach is a machine learning approach, where neural networks are trained to perform tasks such as classification or segmentation. These workflows may produce different interconnected outputs, such as plots, segmented images, or prediction labels. When analyzing scientific images, it's important to prioritize open and free software. These tools not only reduce costs, but also promote reproducibility and transparency. Some widely used tools include Fiji, an extended version of ImageJ, CellPose for robust segmentation, Elastic for machine learning-based image classification, Omuro for image storage and metadata management, ChewPath, specialized in pathology image analysis, Python, the backbone of many custom workflows, NapRE, a Python-based interactive viewer, and platforms like Galaxy also enable cloud-based, shareable image analysis pipelines. A powerful, community-driven resource for image analysis is bioimage.io. This platform provides access to a collection of pre-trained models ready to be applied to microscopy images. These models can be directly integrated into tools like Fiji, Elastic, NapRE, or ImageJ, offering a plug-and-play solution for complex tasks such as nucleus segmentation and cell detection. When sharing image analysis workflows, it's essential to provide clear and reproducible documentation. This checklist from Schmid et al. 2023 presents minimum, recommended, and ideal standards across different workflow types. Key elements include naming the workflow or platform, providing example data, specifying software versions, sharing parameters and rationales, and clearly stating limitations. This quote by Jeffrey Snover highlights a fundamental idea in research reproducibility. The mouse is antisocial. The GUI is antisocial. So what's that mean? You have a problem to solve, and you solve it with a GUI. What do you have? A problem solved. But when you solve it with a command line interface in a scripting environment, you have an artifact. And all of a sudden, that artifact can be shared with someone. That's why we advocate for using code, not the mouse, as a path toward open science, reproducibility, and collaboration. To conclude, let's highlight 3 key recommendations for handling imaging data. First, track all changes and analysis procedures performed on images. This ensures transparency and reproducibility. Second, use consistent naming conventions to link files across the processing workflow. This helps you stay organized and reduces the risk of error. And third, select what to preserve and share carefully, whether final results or intermediate steps, considering storage capacity and long-term accessibility. Publishing your research objects in active research data management platforms, such as the Open Science Framework, OSF, is an effective way to promote open and reproducible science. Don't hesitate to share the illustrative materials you use for your publications, such as figures, tables, and sample images. These materials help others understand and reproduce your findings and are also valuable educational and public resources. Good practices also apply to illustrative images, adding value and enhancing their potential for reuse in different contexts. To gain insight into how to communicate image-based results clearly and effectively, explore real examples of published result images and figures on the OSF via the provided links. Be aware that despite increasing emphasis on open science, data is still rarely shared openly. The common phrase, "data is available on request," has proven to be unreliable in practice. According to open science principles, we should share our data as openly as possible and only restrict access when absolutely necessary, for ethical, legal, or privacy reasons. Sharing data is not just a bureaucratic requirement from funding agencies or journals. It is, above all, an ethical and professional responsibility. By depositing data sets in repositories, researchers contribute to reproducible science and enable the access, validation, and reuse of data by others. To ensure the integrity and impact of scientific work, research must move towards competent researchers trained in research data management and data analysis. Also, standardized approaches for sharing raw data and analysis code to support and verify findings. And last but not least, a strong commitment to transparency, open practices, and rigorous standards to uphold research integrity. For secure, long-term access and sharing of your imaging data, consider depositing in a Canadian research data repository. You can visit FRDR, the Federated Research Data Repository, or Borealis, a multidisciplinary data repository serving researchers across Canada. These platforms support open science and help make your data fair, findable, accessible, interoperable, and reusable. We invite you to explore these resources to support your efforts to make your bioimaging data fair and reproducible. You will find guides on fair image data practices, metadata standards, FRDR documentation, and training resources from the Digital Research Alliance of Canada. The presentation is available in both English and French on GitHub. Scan the QR code to access it. If you need assistance with preparing or sharing your imaging data, please contact us at rdmgdr@AllianceScan.ca or visit FRDR-DFDR.ca.